People have been studying the brain for 100 years, but we still don't have a general theory of how it works. And that is true not just for the human brain, but for the brain of any animal. We're lacking a comprehensive theoretical framework that describes how brains work. Hi, my name is Rafael Yuste. I'm a professor of biological sciences and neuroscience at Columbia University. I'm the director of the Center for Neurotechnology at Columbia. Our research involves understanding how neural circuits work. We are neurobiologists and we use optics. We are also an optics lab. We do nonlinear microscopy, two-photon microscopy, to image the activity of neuronal circuits and to manipulate the activity of neuronal circuits. Although we have a lot of information about individual neurons and about the molecular structure of neurons, and we also have information about the behavior of animals and psychological and psychophysical studies, we're lacking the middle ground, which is to understand how neural circuits work. What is a neural circuit? A neural circuit is a group of neurons that are wired together, forming an assembly, and it is very possible that it is the actions of those assemblies of neurons, those neural circuits, which are the ones that are generating behavior or mental states. We have methods that allow us to study single cells with electrodes, and we have methods that enable us to look at the entire brain at once, like with magnetic imaging methods. But we have this big gap of knowledge in watching how hundreds of neurons or thousands of neurons interact together and to form functional units. So the work that we do, the methods that we develop in the lab, are aimed exactly at that middle ground. Can we see the activity of every neuron in a neural circuit? And can we manipulate the activity of every neuron in a neural circuit? Because if we can do that, we may be able to put the missing brick in the building and enable people to build a general theory. In my career, we're trying to tackle this basic understanding of neural circuits as our main goal. In particular, we're interested in the cerebral cortex of mammals, which is the larger part of our brains that is right under the skull. It covers our encephalus. And that's the region that is damaged in mental diseases and many neurological diseases. So we're focusing the fire there to try to come up with a basic understanding for how it works so that we can then apply that knowledge to try to understand disease models. In our lab, most of the people are working on basic questions, fundamental science, but there's a few people that are studying mouse models of particular mental or neurological diseases. So there's a person working on mouse models of schizophrenia. These are mice that have some of the same problems that schizophrenics have. And we also have someone working on a mouse model of autism. You can actually build genetically engineered mice that resemble in some of their phenotypes autistic patients. And finally, we have a person working on a mouse model of epilepsy. We can generate epilepsy artificially in a mouse and study it to try to understand what is the problem with the epilepsy in human patients. So, we're very convinced that through the introduction of these methods, scientists are going to be able to actually finally get to decipher the neural systems, break the code that the brain is using to communicate with itself. And this can be used not just for scientific understanding of how the brain works, but also for clinical understanding of the diseases that patients have, the pathophysiology of the diseases, or the design of novel therapies. But we're still in the very early days. You have to realize that the human brain has 85 billion neurons, and the methods that we have today, even in the best labs in the world, even in a lab like my own lab, we can at most read out the activity of maybe 1,000 neurons and manipulate the activity of maybe 100 neurons. So it's a very far cry from being able to systematically gather the information from an entire neural system, even in a small animal. it gets worse. These methods that we need have to work in 3 dimensions because the brain is not a flat surface, it's a 3-dimensional substrate. But the microscopy traditionally developed is always focused on the focal plane. It's always a 2-dimensional method. So part of what we have to do is to reinvent microscopy and turn it from a 2D method into a 3D method, and also come up with clever ways of going into the brain of animals or humans and alter the activity of neurons. And we think this can be done in principle with light.